So for our research, Dr. ORCUTT has traveled far and wide above the ocean, exploring underwater hydrothermal vents, oil seeps in Gulf of Mexico, and research labs around the world, including a recent sabbatical in Germany. Beth is also a senior scientist at the Center for Dark Energy Biosphere Investigations, which is a NSF-funded science and technology center. And she was recently awarded the Asaiko Taira Prize, I hope I said that well, for scientific research from the American Geophysical Union through her implication and work within the IODP program, working on drilled boreholes to study subsea floor microbial life. So I'm delighted to host you today. Thank you very much for accepting to give this talk, and the floor is yours.
Great. Thank you so much for the invitation to join the seminar series. And thank you to everybody that could tune in live today. I look forward to hearing your questions later. If you don't get a chance to ask me questions in today's session, or if you're watching this as a recording, feel free to follow up with me either on Twitter or via email. And I'm excited to talk to you today about thinking about life on rocks at the bottom of our ocean. But before I begin, I want to acknowledge that where I work in Maine is on the ancestral homeland of the Wabanaki Confederacy of Native American groups. And I'm trying to think about how I can bring decolonialization concepts to my work. And I welcome any questions about that as well. And I also want to start by thanking the funders for this work. A lot of the science I will present to you today is enabled by the International Ocean Discovery Program, the Scientific Ocean Drilling Program, and has been supported also by the Sloan Foundation funded Deep Carbon Observatory, funding from NASA, from the National Science Foundation, and as was just mentioned, the Center for Dark Energy Biosphere Investigations, or CDEBI. Okay, so let's begin. Hopefully I can get this to work. So when I like to think about our seafloor, and if you strip all the water away, I think maybe Gretchen introduced me to an image like this. What I find fascinating is just the wide variety of seafloor habitats that we know very little about, right? There's all these underwater mountains, the volcanoes, subduction zones, etc. And all of these places could potentially be habitat for life on Earth. And in this map view, you can see that there's more of that seafloor than there is of land. So if we really want to understand life on Earth, we should have a better understanding of life in the deep sea and the seafloor and the subsea floor. In a cross-section view, I take this cartoon from an annual review that Matt Schrank and Julie Huber and Katrina Edwards had put together some time ago, kind of illustrating some of these different habitat types. And if you were to focus on a mid-ocean ridge section and thinking about, there's vertical structure in the ocean crust, where you can have extrusive basalts near the surface and more dike-like formations below that, as well as gabbro or even mantle type rock deeper down. And so along the mid-ocean ridges, you have new ocean crusts being formed as plates pull apart and are pulled down through subduction. You can also have seamount systems that are off-axis, as well as the volcanic back arcs and volcanic arcs in those subduction zones. So many different types of habitats where water-rock reactions can be happening that support life. In a very simplistic view, and my apologies to any chemists who are like, what? I think about this in the sense of when either basalts or mafic type rocks or ultramafic rocks react with the seawater, some of the byproducts of the reactions are chemicals that microbes can use to live. So for instance, in mafic settings, you can have hydrogen sulfide or reduced iron in those systems that provide energy for microbes. In ultramafic settings, some of those byproducts could be hydrogen and methane as secondary byproducts of those reactions. And so through these fluid rock reaction processes, you could be generating substances that microbes can use. And so when I see a rock like this that we collect through ocean drilling, and I think about these veins and the secondary minerals that are precipitating, what I want to know is, like, where are the microbes in those rocks and who are they? And are they potentially speeding up these reactions? or slowing them down. And as I come towards the end of the talk, we'll think about why that's important. But first, I want to make sure that we're all on the same page about what I mean when I say the word microbes, because I know we have a very diverse audience tuning in today. So really, this is an operational definition based on whether or not you can see the life with your own naked eye, or if you need a microscope to see it. And so microbes could come from a wide variety of life. They're not all just bacteria. They could also include the domain Archaea. They could include small fungi. They could include viruses. Throughout most of this talk, when I say the word microbes, I'm really referring to bacteria and Archaea, because I particularly don't study viruses or microscopic eukaryotes myself very often. But I'll try to clarify that throughout this talk. And the reason that I focus so much on bacteria and Archaea is because they can do so many cool reactions that are important for global chemical cycling. So this table, very detailed, I don't expect you to memorize it. It's just a listing of all the different types of, or some of the standard chemical reactions that microbes are involved in. Whether that is a very simple type of process that we're all very familiar with, which is respiring oxygen to burn carbon, like sugars and fats and proteins that might be eaten. But microbes can also breathe other things beyond oxygen. They can use nitrate as an oxidant to perform redox reactions. They can use oxidized metals. They can use sulfate. There are organisms that can generate methane. So this is really how I got interested in thinking about microbes in the deep sea, is by thinking about the chemistry that they might be involved in, and either speeding up or slowing down some of these processes. So in a broad view, I think about the deep sea in kind of different types of habitats. And I show here a cartoon from a recent book chapter that isn't in the book isn't published yet, but it's out as a OSF preprint, so you can find it if you want it. So if we were looking at the seafloor, the kind of habitats that I focus on, or that I will focus on in most of my talk today, range from hydrothermal systems, where you have on mid-ocean ridge and the flanks of mid-ocean ridges, to the seamount systems, where you have maybe environments that used to be on a mid-ocean ridge and have now moved off axis and have been surrounded by sediment, or on a hotspot where seamounts are created, as well as on the abyssal plains, where you might have the formation of manganese nodules or polymetallic nodules that form over geologic timescales out in the abyssal plains. In all these environments, there are microbes, probably performing very important processes, although the extent to which we know them is relatively minimal, as I'll show. And in the work that my team does, we both look at seafloor processes as well as subseafloor processes. So if you look to the right-hand side of this image, you can see what looks like a drill ship going into the subsea floor to understand life in the ocean crust. And the most of my talk today will focus on those types of environments, but I'll touch on all of them. So to start with, part of the reason one of the ways that we frame our thinking about microbes in the subsea floor and on the seafloor is in terms of the services that they provide. This is kind of a weird definition that's used a lot in conservation space to think about the importance of habitats and what habitat services are provided and how they might be impacted by human activities. And I've started thinking about some of these processes in that framework because of potential human impacts that we'll come to later in the talk. So if you were to zoom in on any one of these habitats in this cartoon, you might see that there's microbes living on the rocks themselves, and those microbes can be involved in different things, like for instance, taking carbon dioxide and converting it into organic matter. And that would fall under one of these provisioning services called primary production. So different microbes in the environment might be taking carbon dioxide, converting it into organic carbon that then other life forms in the ecosystem can use. As another example, microbes can also perform the service of transforming organic carbon and taking it from a more complex form into a simpler form that other organisms can use. Another type of ecosystem service that falls under more of a supporting category is the important nutrient cycling and metal cycling that microbes do. So for instance, there are some microbes that can take up ammonium and convert it into nitrate, which is more bioavailable for other life. They can also take reduced iron and convert it into oxidized iron. And the involvement in metal cycling can be important because all life needs metals for our enzymes to work. And so they can play an important role in that. There are other types of services that microbes might be providing in these ecosystems, but that are not really well characterized. So things like maybe microbes are producing certain compounds that signal to animal larvae that this is a good spot to lay down and make another generation of your species. But it's not really well characterized. And in many ways, most of these services are not well characterized. So I put here kind of a question mark to emphasize that while we know that these things may be occurring, often we don't know the magnitude that they're occurring at, who might be involved in these processes, and how they might respond to perturbation. So there's a lot of kind of unanswered questions in the deep sea in thinking about these, the role of microbes. When I got started as a postdoc working with Katrina Edwards, in my first kind of exposure to thinking about life on rocks, I was involved in a project led by Brandy Toner that wanted to look at all kinds of published data sets as well as some new data sets from the East Pacific Rise, and ask the question, does the structure of microbial communities living on seafloor rocks, does it matter what the mineral is or the rock type is that they're growing on? If you ask it another way, do certain kind of rocks have certain types of microbes? And so in that study that came out in 2013, where the state of the art was looking at just one particular gene from an environment that was generated either through a process called clone libraries, maybe a little bit of next generation amplicon sequencing that is all very common today. A decade ago, those were not very common. And so the data sets were pretty sparse. But when we looked at that, and this was work that Ryan Lesniewski did as a student involved in this project, was comparing those different types of ecosystems and looking at them. And in a very broad sense, what we could see in that analysis is if we created a dendrogram looking at the similarity of community structure on different types of samples, and we grouped them by whether they were from seawater and sediments, whether they were like a barium-rich chimney hydrothermal system, an active hydrothermal chimney system, inactive sulfides or basalts, which is kind of covered up here, but it's green. In a very broad sense, Ryan's analysis indicated that there is structuring of microbial communities that seems to be related to the rock type that they're on. So that was kind of a question that we had then, but we wanted to explore that in more detail as time has gone on. So how do we do that? For anyone who is unfamiliar with the ocean drilling program, I wanted to make sure that I included a schematic to show you how we do this. So this is really the flagship of the ocean drilling program, the Geordes Resolution. And the way this works is that in this contraption that looks like an oil derrick, a drill pipe is assembled and lowered down to the seafloor that allows us to go through either sediment if it's there and into the hard rocks below. And then we bring those rock cores up onto the ship and try to figure out who they are. And so this is a picture from an expedition in 2011. I'm not sure if Wolfgang is on the call today, but if he is, there he is, with me trying to decide which samples should we look at for trying to understand the microbes from these very precious subsea floor samples. The way that we try to do this is we take a rock sample, we want to figure out who the microbes are, so we use an approach where we extract all the DNA out of that sample. And then we try to look at taxonomic marker genes in that DNA and put it within a known tree of life. And this is a very old tree of life from the late 90s, but it gives you the general idea that you have different domains of bacteria, archaea, eukarya, and we're trying to figure out where do our microbes fit in this tree of life. This is really difficult. So the rocks that we collect in the drilling program often come up contaminated with seawater, with drilling mud, people touching them, et cetera. So we have to use really aggressive methods to try to get off all that contamination from the rock samples, to get at the microbes that might be within the rocks. And this is challenging. It is not a trivial matter. One of the things that we have done recently over the last decade or so is taken advantage of some borehole systems that are left behind after ocean drilling and installed instrumentation in those holes that allow us to study microbes in the rocks and in the fluids that move through those rocks over time by either putting in sensors or putting in different experiments, et cetera. And so these are cartoons and schematics to kind of show what that looks like. We're trying to get at those microbes living on the rocks in a way where we can allow the system to reset and recover from the drilling disturbance. And so the way that we do that with the groups that I have worked with is by deploying these colonization experiments inside of those boreholes. And so these colonization experiments are basically flow through reactors that we pack with either crushed up minerals or with polished mineral coupons ranging from basalts to metal oxides to metal sulfides to relatively non-reactive substrates like glass or plastic. And we put them in those holes in areas where the borehole observatory has slots in it to allow fluid movement to go through the well. And so microbes that might be in the fluids, in the subsurface, can come through and colonize our sterile rocks. We can leave them there for several years and then recover them and see what's growing on them. So here's just an example of what that might look like. Take one of those polished coupons, we can put it under an SEM microscope and we can see that there's microbes that have grown on them. And so I want to tell you about a couple different expeditions where we've done this type of work and what we've learned. So there's three sites that I'll mention in my talk today. One of them is on the off the west coast of North America on the Juan de Fuca Ridge flank. Because this site is so close to land, it gets buried in sediment coming off of land. And so it's relatively warm compared to most ocean crust in a seamount system. And because it's warm, it also means it's lost its oxygen and it's relatively reducing conditions compared to what we think most ocean crust is like. I'm going to compare that with the North Pond site, which is on the western flank of the Mid-Atlantic Ridge, which is more representative of what we think global ocean crust is like. Cold and having oxic fluids moving through the system. And then I'll end it by saying what we've been learning about from an ultramafic system that's also on the Mid-Atlantic Ridge at an underwater mountain called the Atlantis Massif, which is also host to the Lost City hydrothermal vents, which you may have heard of. So first, starting with that Juan de Fuca Ridge flank environment, I want to tell you about an experiment that we started in 2010 and recovered in 2014. And this is work that was led by Gustavo Ramirez, as well as Aurélien Lecouvre, who was a visiting master's student in my group at that time. So for this experiment, what happened is we deployed these colonization experiments, both in the subsea floor, where the conditions were warm and anoxic, and we had another kind of parallel experiment that we deployed at the wellhead of the observatory. It's accessing the same crustal fluid, but by the time the fluid has gotten up to the top of the observatory, it's cooled down. So it's more like the temperature of your refrigerator or cooler. So we have kind of two different comparisons, and then we have basically two rock types that we've been looking at in these experiments, some basalts, as well as pyrite and pyrrhotite. And again, the general concept of these experiments is that within our flow-through systems, which we call flocks, the fluids that are in the crustal fluid, the fluid that is circulating through the rocks has microbes in it. And those microbes may represent what's grown on the rocks, but it could also be planktonic microbes that are just kind of floating through the system. They get pulled into our colonization experiment, and hopefully they'll start growing on the rocks that we've provided. And so we can look at both the microbial communities that have grown on our coupons, as well as what might be been in the rocks that we've drilled, as well as in the fluids themselves. And so in this paper that came out a little over a year ago now, what we show here is that when we extract DNA from our samples, and we look at the composition of the microbial community as a percentage from 0 to 100 along the x-axis here. You can see by eye that there are differences in the structures of these microbial communities where each one of these rows represents a different sample. Now you will notice here I did not include a legend, because for the point of this story, who the microbes are is actually not that important. And I don't want to give you a word soup of microbe names. What I want you to take home from this data is that the experiments that we had under the cold conditions at the wellhead have a very different microbial community structure than what was growing on the same minerals in 64 degrees and under anoxic conditions. You can also see that by looking at the names of the rows, that whether or not there's a difference based on whether it was a basalt or a pyrite isn't really that clear. So the end result of this experiment is that temperature and maybe redox plays a really important role in the structure of microbial communities that grow on the rocks, but that the minerals themselves may not play as important of a role. We'll come back to that in my next example. Okay, so again, we're gonna keep that in our mind. We have the Wanda Fuka experiment example. Now we're gonna go to the cold and oxic system at North Pond and look at a similar type of experiment we did there. So to introduce you to this site a little bit more, the North Pond location is a small little pond of sediment that is formed in the valley of exposed outcrops on the western flank of the Mid-Atlantic Ridge. And we visited the site in 2011 and cored some new holes through the sediment to get into the rocks below. If you were to look at that site in cross section, here's kind of how we envision it. So you have this small little pond of sediment that ranges from about 40 to 90 meters thick in the valley between all these outcrops. And we installed a series of borehole observatories within this ocean crust setting. Some of those boreholes only went very shallow into the ocean crust. Some of them went deeper into the ocean crust, and we were actually able to seal off different horizons with packers and other mechanisms. Not all of the experiments worked. Doing deep sea work is very challenging. So in the schematic, things that are in gray, we were not actually able to recover. And so for this talk, what I'll be focusing on are some colonization experiments that are shown in green here. So we have one experiment that was deployed at a site called 1382A into upper oceanic crust. We had another very similar experiment deployed at U1383C. And then we had a similar experiment deployed in a cork light observatory at the same site. And so for this experiment, literally thousands of colonization experiments were deployed, and we looked at a few hundred of them for this study. And so just to show you kind of what they look like going down and coming back, on the outside, we had these polished mineral coupons, and on the inside, which you can't see very well, are some of those packed, crushed up rock types. So again, same concept, you deploy these in the subsurface, and we were looking to see what microbes grew on them that were colonized or inoculated by the crustal fluids that are moving through the system. So these were deployed in 2011 and recovered in 2017. So they were incubating in the subsea floor for 6 years. And we learned a lot of things in this study, but what I want to focus your attention on is just this one graph. So this is a graph in a recent paper that came out just within the last month within environmental microbiology. And I want to give a lot of credit to Tim D'Angelo, who did the majority of this work. What Tim did is extract DNA from all these different colonization experiments. We sequenced the 16S rRNA taxonomic marker gene with Illumina sequencing, and we compared the structure of all the microbial communities on these rocks to each other. And so what you're looking at here is kind of a relationship graph where the closer symbols are to each other indicates that the communities on those samples were relatively similar in terms of the types of microbes that were there and the relative abundance of those microbes. And in this study, we're doing our experiments at 3 different sites. So the color of the symbols here indicates the site where the samples came from. And then the symbol shape is an indication of what the underlying sample was. Was it a basalt? Was it a metal sulfide? Was it a metal oxide? So what you can see by eye in this graph is that the symbols group most closely to each other based on where the experiment was done. And there's not a clustering of these symbols based on the mineral type. So for instance, all the star-shaped symbols aren't together, or all the square-shaped symbols are together. So what that tells me is that in this experiment, the type of rock that the microbes were growing on didn't really matter. What mattered was the fluid that they were being exposed to. So on the time scale of this experiment, on a decadal time scale, the structure of a microbial community doesn't really depend on the mineral type. This is maybe a bit counterintuitive to what you might expect. You know, a metal sulfide will be much more reactive than, let's say, a basalt. And so while there may be individual members of the community that do respond to the, for instance, differing reactivity. So for instance, on these metal sulfides, we do see some organisms that are known to be involved in sulfur cycling. But overall, the overall community structure doesn't really seem to matter, which may indicate that the microbes that live on rocks are more generalists in their approach. And so this is something we want to keep digging into. This is relatively fresh data. Another thing I want to point out to you about this site is a recent work done by Jeff Wheat and colleagues on that 2017 cruise. Jeff and his team came up with a way to try to do a crosshole tracer experiment, where we went to one of the holes and Jeff and his team injected a tracer solution, in this case that was doped with cesium, and we were able to detect that tracer show up in another borehole observatory roughly 35 or so meters away within days. So that means that this site with these observatories is primed for doing some really cool experiments to look at, if you inject things into the subsurface, what happens to them as fluids move through the system. Really cool paper. I encourage you to have a look at it. Okay, and then the third example I want to focus on is some recent data from the Atlantis Massif. So again, this site's a little bit different. It's not necessarily a mid-ocean ridge system with lots of basalt. It's actually a deeper mantle type rock that's been subsumed to the seafloor. And there's many on this call who know a lot more about this process than I do. In 2015, we used a mission-specific platform of the Ocean Drilling Program to go to this underwater mountain and drill a series of sites on the southern wall of the Massif with the goal of trying to figure out what kind of microbes live in these serpentinizing systems. Again, on the left here is a map showing you these sites along that southern wall with the velocity site for reference. We didn't get as deep as we would like on this core expedition, but we got some really amazing core back, really high recovery. Even, and going from things like baby powder, talc, which is very difficult to drill, to very beautiful serpentinized materials. There's a great study summary done by some of the team of the Atlantis Massif showing the degree of serpentinization in the system and talking about the evolution of the system. I'm not going to go into details here, but I wanted to put up the reference here in case you wanted to look. But just keep in mind that we recovered some really great serpentinized materials from this environment. And so our question then, or my team and my collaborators, was what are living on those rocks? Are they similar to what we find in basaltic systems? And so we really tried to focus our attention to our methods to make sure we were doing this as cleanly as possible, using, for instance, frozen diamond tip saws and working in a coach system, which you can see Yuki Muro on the right. And it's really important that we did this because if we look at the cell densities of in these rock samples, they are very, very low, like on the order of 10 to 1,000 cells per cubic centimeter. This is really pushing the limit of detection of cells in these environments. And so kudos to Yuki for generating this data. But what this means is that even those standard approaches of trying to extract bulk DNA from these samples is going to be extremely difficult, if not impossible. There's a recent paper out led by Sherry Ortmedi from Billy Brazelton's lab that goes into this, and the blood, sweat, and tears that went into trying to get data from these samples using that approach. My team has been trying a different approach. So before the pandemic and wearing masks everywhere was very fashionable, we were using this type of approach for trying to get at single cell based sorting. So taking one of these rock samples, trying to liberate all the cells off of it into a fluid solution and putting them in a flow cytometer where you could sort out individual cells and using the facilities of the Aquatic Cytometry Center and the Single Cell Genomics Facility at Bigelow Laboratory. And I won't show you much data because this is work in progress, but I just want to show you it works. So this is an example flowgram of an Atlantis massive sample. What you're looking at here, all the colored particles indicate individual cells that we were able to sort and amplify the DNA of and create partial genomes for. And the color represents the different groups of organisms that we've been finding. And so stay tuned. Jackie Gorgel is working on that paper right now, and we hope to have it out to you soon so you can learn what we're finding. Okay, so I've talked to you a bit about how we do this, but now I want to switch our attention to like, why do we care about this? Because there's a really important things on the horizon in terms of thinking about mid-ocean ridge systems and coastal systems in general that I think we should be talking about more. So a lot of the work I do is really motivated by this idea that life in the subsurface really gets at extremes of temperature, pressure, carbon availability. Like it allows us to study the fringe of life. This graphic here from the recent Deep Carbon book, I think explains that well, that we're really looking at those fringe environments. And that's important to me because I like to think about the idea that life might exist on another planet. And that's why NASA has given us some funding to do this kind of work. And while those basic motivations are important, we also need to think about the other aspects of why this work is important. So I want to come back to this cartoon that I showed you at the beginning and think about some of these habitats and some of the emergent human uses of these ecosystems. And those are kind of represented with some of these cartoons in the upper portion where you can see these little vehicles. And why is there interest in this? Why do humans want to come into these environments? That's because a lot of the rocks on the seafloor contain metals in relatively high abundances that if you were to find these same abundances on land, might be considered an important ore. So for instance, here's an example of the average content of some manganese nodules in particular areas and how much cobalt, copper, et cetera, are in these systems. May not seem like a lot, but economically, this might be viable to go get. And as you all know, there's a real movement around the world to move towards fossil-free futures, which is great. But all of those technologies require metals, whether it's solar panels needing metals, or windmills and electric vehicles, et cetera, we're being reliant on lithium alloy batteries. And so we need to understand there's a crazy demand for some of these metals right now because of this push for based on consumer use of metals. And so there is an interest in exploring some of these deep sea environments for the concept that they might have metals. And there's a lot of discussion right now about whether this is a better strategy than, for instance, mining on land to get some of these metals. So there are 4 different habitat types that are thought of for these environments for metals. Those are, again, the abyssal plains where manganese nodules form, at mid-ocean ridge settings where you have hydrothermal sulfides being generated, as well as at seamounts that have been exposed to seawater for a long period of time and have had extensive alteration crust form on those rocks, which are often referred to as cobalt crusts. So I'll zoom in on a few of these and give you some examples of what we know. The best studied site really for understanding potential impacts is in these nodule fields in the abyssal plains. So we'll focus on that first. When I was a PhD student, which I won't tell you how long ago that was, this was presented already as an idea, but it was presented in kind of like a sci-fi context kind of way. with, just cartoons and, but not really as this is viable or going to happen. But that conversation has really changed over the last 5 to 10 years, where there are actual prototype vehicles that are being tested, and the seafloor is being contracted out for this kind of exploratory work. So if we look at a map of the seafloor and think about where these types of resources are found, there's, again, kind of 3 main resources. There's those nodules that form in abyssal plains, which generally are underneath the mid-ocean gyres. The cobalt crusts, which form on seamounts. There's many of those in the Pacific. And then the hydrothermal sulfides, which are concentrated where you have active mid-ocean ridge processes along mid-ocean ridges. So again, the nodules are kind of the poster child right now of this kind of work. And we're going to focus in on a site called the Clarion-Clipperton Zone, which is out in the North Pacific. And just keep this in your mind, the scale here, that area that's highlighted with a dashed line, that's the Clarion-Clipperton Zone. Compare that to the size of the continental US in terms of how wide it is. So if we zoom in on that area, this is the Clarion-Clipperton Zone. As of 2018, there were 15 different exploration contracts to go into this area with manganese nodules to see if it might be viable to mine these resources. At the same time, looking at these environments, there's a lot we don't know in terms of what species live there, what is the diversity of life there. So there's been some really great studies out in the past few years trying to catalog this diversity. I just show one example here that cataloged anemone and sponge and brittle star, for example, diversity. Some of these species being new to mankind in terms of our understanding of who they are. And there's been just a few studies to try to understand what the impacts might be to mining type activities in these environments. Again, pointing you to this study that came out in 2016, where an environment that had manganese nodules had kind of like prototype mining or test, not mining, but looking at what the potential impacts would be of dragging a dredge through a manganese nodule field as if it were going to remove manganese nodules in a mining type activity. That site was revisited 35 years later to see, like, had the system recovered? So in the top left-hand corner, you can see a sample, an example of what unaltered nodule fields looks like. You have all these little potato-shaped lumps everywhere. You've got life there, indicated by the purple sea slug and some other guys. On the right, you see an example of what unimpacted seafloor looks like that doesn't have a lot of manganese nodules on it. Now, what I hope you can see is that there's little trails through it indicating animal life moving around. If you compare that to the area that had been previously dredged, there's no animals there. You don't see any indication that animals have been there. And so within the time scale of several decades, doesn't seem that there's recovery. This was just looking at animals, and there's been a more recent study done by colleagues in Europe, looking at this from a microbial standpoint. So a similar type of experiment where a dredge had been pulled through a system, as well as recent dredging done on the expedition, and looking at whether microbes in these systems were impacted. And so the color scheme here, green indicates unimpacted nodules, orange indicates areas that have been impacted decades previously, I think 26 years in this case, and then red indicates just recently, within weeks impacted. And I just show here one example of the data that they recovered looking at primary production by microbes. So the fixation of carbon into organic carbon. And you can very clearly see by eye that even in systems that have had 26 years to recover, they still show signs of impact. They are not performing the same service to the same magnitude as in an unimpacted setting. So that really indicates to us that these impacts could be very long lasting in these ecosystems if we choose to do them. Hydrothermal vent systems, so switching away from manganese nodule fields and thinking about hydrothermal vent systems. Again, these are generally located along mid-ocean ridges. I'm just going to zoom in on one in the North Atlantic, where this is on the northern mid-Atlantic ridge, south of the Azores. There are currently 3 contractors, Russia, France, and Poland, that have permission to explore these environments for potential mining. And just to highlight to you, known hydrothermal vents that scientists study are in those same contract zones. And so there's active conversation happening right now with the International Seabed Authority and among science groups like the Deep Ocean Stewardship Initiative about how could mining potentially happen anywhere near these sites and not impact the unique communities that live at these hydrothermal vent systems, which I think have been topics of other Ridge Symposium talks, so I won't go into those. I want to end here with thinking about the seamount systems. So in my perception of the deep sea environments that could be impacted by mining, we know the least about microbes and the role that they play in these ecosystems. And the reason that this is important is, hopefully this video will work. This is a video taken at the Dorado Outcrop Seamount, which is in Costa Rican waters on the Pacific side, where I was part of a team looking for evidence of low temperature fluid flow at seamount systems. And in our work there, the team discovered that octopus are totally colonizing these seamount systems and laying their eggs where this low temperature fluid flow is occurring. This phenomenon has been now observed at other sites, like in the Monterey Bay system, of these dense octopus gardens, and trying to understand why they're laying their eggs in these systems. But it's just an example of, like, we didn't know that that behavior existed. And now we don't know if that behavior is widespread, if the low temperature fluid flow is widespread, et cetera. And so and you can see that these rocks are heavily altered. They could have these metals in them. And so is it worth protecting environments like this because of the unique animal behaviors that are observed there? And the final potential human impact that is on the horizon, I think we need to be thinking about when thinking about microbes and life in the subsurface, is this idea of potentially taking advantage of the fact that carbonate precipitates in basaltic subseafloor settings and accelerating that process as a form of carbon sequestration. So I borrowed this graphic from Solid Carbon, which is a team based in Canada that's working on this concept of, could trapping carbon as in form of carbonate in the subsea floor be a way to sequester carbon as a climate change mitigation strategy? So hopefully I've convinced you that you should care about life on these rocks and trying to understand this. I'm trying to work with a team to try to accelerate some of this work, because there's really a lot we don't know, again, about the magnitude of the ecosystem services, how resilient the microbes are in these systems to perturbation. And because of the emergent human uses, I'll just pull up all of this. We need to have a better understanding of this to inform potential commercial or industrial use in these systems. And so we have this concept that we put forward called COBRA, for the Crustal Ocean Biosphere Research Accelerator, to try to help coordinate research, because there's lots of groups thinking about this, but we could be leveraging our assets better to accelerate that characterization, and to really think about how we translate what we're finding for policymakers as well as industry, and to be mindful that the assets to study these systems are not equally distributed, but the people that are going to be impacted from this are globally distributed. So how can we better leverage the resources we have to build a future cohort of ocean leaders and stakeholders that have access and can drive this research? So I'd be happy to answer questions about this initiative, as well as any of the science. And with that, I'm going to thank everybody that has been involved, and hopefully you are well aware that we love rocks and the microbes that live on them, and I welcome follow-up conversation. And I'm going to end my slideshow so now I can actually see who is there. Thank you so much.